right, a lot of questions have come in, sir. Thank you so
Sure.
much for those wonderful talk. So one of the most important questions which is going around is that you talked about the concept of molecular tracing, realizing that how a majority first came in from outside and now we have a sequence which is probably indigenous. But before that, just one brief clarification. Are these mutations sufficient enough to cause by themselves a new strain, like how traditionally we have been taught the concept of antigenic shift and antigenic drift, so that it completely changes in its nature? Or are these mutations causing something else? I mean, what is the terminology that we would probably use when we see this?
Yeah, so to be accurate, there is only one strain, and that is SARS-CoV-2. And this is to differentiate the strain from the earlier SARS epidemic, which happened way back almost a decade and a half ago. So theoretically, there is only one strain, that is SARS-CoV-2, and all that is genetic clades that are formed by mutations are essentially lineages. So they can be called as genetic lineages, or they can be called as clades of the virus. So theoretically, there's only one strain. It is not multiple strains of SARS-CoV-2. They're only genetic lineages of SARS-CoV-2.
I think that's a very, very important point to put across to everyone out there, that there is only one strain of SARS-CoV-2 and all these mutations result in probably different genetic lineages, or the word, as you have rightly put it out, is known as clades as such. So now the second question that is coming in is that now the world is talking that it's a completely different mutation that we are seeing in right now, which is the mutation being called as the D614G mutation, and that is probably more infective. Sir, according to you, what is the current genomic makeup in India in terms of the percentages of the different mutations which is there? What is the most common clade that we are seeing here?
Yes, so the most common trait, as you see here from this figure, though this is a bit outdated figure, it still holds good. Most of the isolates from India belong to the A2A clade, and the A2A clade is marked by the mutation D614G.
Okay.
And this is not just in India, this is also globally the most predominant clade, because as you rightly said, there is enough experimental evidence today to suggest that this particular mutation latches onto the human receptors much better, and therefore they could have a much higher infectivity compared to mutations which don't have it.
All right. So there's a question coming in from Dr. Tarun Sebastian. He's a gastroenterologist and hepatologist at Rajgiri Hospital, Kochi. So his question is that at least from data which is available, there is a slightly higher mortality rate among healthcare workers. Now, the hypothesis or the question is that could it be because of the presence of multiple strains simultaneously? So would the current platforms of testing be able to detect multiple strains present simultaneously?
So yes, we did. So the sequencing allows you to identify the haplogroups and also multiple strains which might be present, or multiple viruses which might be present at the time of infection. And but multi-sample infection or multi-clade infection is something that is extremely rare. We have not really found any such case yet in our data set. So I would not really subscribe to the idea that clinicians get multiple viruses infected at the same point of time. And I will also not probably subscribe to the idea that clinicians have a higher mortality rate compared to the normal population. Probably if you really go back to the statistics, clinicians have much lower mortality compared to the general public. And of course, there are multiple reasons to this. One is that they are aware about the epidemic, they take much better precautions. And of course, they're also what you call aware enough to seek healthcare when they need the most. So it is not really true that clinicians have a much higher rate compared to the normal in terms of mortality. But yes, having said that, mortality is mortality, and we really should not go back by statistics. Every single individual is precious, and therefore we should do everything possible to save those individuals.
I mean, absolutely, sir. And I really hope that our policymakers and those who are taking decisions have the philosophy that you put, that each life is important and one death too much, even if it's one, is too much. So there is one more question coming in from Suresh Reddy, who asks, what are the chances of the occurrence of a deadly mutation? In other words, I think what he means to ask is, do you predict, given the previous knowledge of the other coronaviruses, that there is a chance that antigenic shift can occur like we see in the influenza virus?
Yeah, so the fact is that we understand very little about the immunological properties of the virus. Increasing evidence suggests that death is because of overreaction of the immune system, or rather improper reaction of the immune system rather than overreaction. And the improper reaction is a factor of the quantity of response as well as the time at which the response occurs. Now, the real question is, does the mutation change the virus so fast that it can create an antigenic shift? Until this point in time, we don't really know, because we don't really understand what are the regions in the virus that elicits a deadly immune response. There are a couple of new papers that have really come up in the very recent weeks, which suggests very specific epitopes in the virus which could elicit a subset of the hyperactive immune response, the multisystem inflammatory syndrome, especially in children. And this came in PNS a couple of weeks ago, which suggests a very specific motive that causes it. But of course, there is much more to be done, because I don't think a significant amount of the multisystem inflammatory response could be described by just mutations in the virus. So it is also going to be interplay between the virus and the host, because the host genetics is also as important as the viral genomic changes, because you need to keep in mind that if the host needs to respond to a particular virus, it also needs to have these peptides bind to the B cell and the T cell receptors, and to bind to them, of course, they need to be presented appropriately by the MHC molecules. So there are genetic variations in each of these components, which are also quite important, and at this point in time understand so little about this genetic interactions, and hopefully more time and more research would kind of solve many of these dilemmas.
I think that's a very interesting point you make, sir, about the host genomics, because I think recently there came an article which talked that genes which are inherited, or rather a genomic sequence inherited from the Neanderthals, are the one which probably makes certain humans more susceptible to a COVID infection. So there is one, I mean, I'm just trying to club few questions which are of the same type. So now we have certain questions which focus on what you mentioned about the impact of mutation on RT PCR. So I have a question from Dr. Brinder from UDSC and Dr. Kaushik, who are asking that, do you think right now there should be guidelines made to say that, or rather a disclosure that the RT PCR is using this and this primer and probe, and this is what is going to be applicable in testing for these strains of coronavirus? And as a follow-up question for that, considering the mutations, do you think that there could be more false negative in RT PCR, and consequently would antigen be a better test?
Yeah, so absolutely right. So the biggest problem today with most of the RT-PCR kits is that they don't disclose what primers and probes that they use, and which I believe is something not really proper, especially in this particular pandemic. Even to collect the information for primers and probes, we wrote to close to around a couple of hundreds of companies, and the responses which came back are in dozens. So many of these primers and probes that the companies use in the kits are not really disclosed in public domain, which I think is not proper, and that therefore there should be guidelines, because without evidence you cannot really make a decision, right? If you don't know what a kit uses, how are you supposed to make a decision on whether I should use this kit or not use this kit? Now, second, of course, the regulatory agencies should take note of it, and I think it was not be a big problem in the past, but now some of these genetic variations have become much more frequent, when I say frequent, that means more than 2 or 3%. Some of them have actually reached 5% in the population. And then therefore, those reagents or those primers should probably not be used, because then you would have a lot of false negatives. Now, the second question is, are there large number of false negatives? The probable answer is no, because RT-PCR doesn't rely on just one primer or probe. Today, the guidelines suggest that you need to look at two genes, either the RDRP or the E and the M genes. And then therefore, the chances of false negatives using two genes in combination are going to be much less. So it is not like it is an alarming situation that we need to act upon currently, but it is a situation where you have evidence which can make your testing much better.
And so that we are also fortunate to have with us Dr. Ullas PT. He's from the National Institute of Virology. So his question to you, sir, is that has the existence of viral quasi-species challenged their characterization by NGS, and have you experienced this with the SARS-CoV-2?
Yeah, so viral quasi-species is a very interesting phenomenon, and you do have viral quasi-species in SARS-CoV-2 also. The interesting thing is that you can identify quasi-species only by deep coverage genome sequencing. There is no other technique which can allow to do that in an unbiased way. So yes, SARS-CoV-2 also has viral quasi-species, and if you really go back to the data sets, it's a well-documented fact.
So now we know that, I mean, there's a very interesting question coming in from Naga Vaishnavi. We know that the Nobel Prize in Chemistry went to the CRISPR, the people who discovered CRISPR. So she's asking, is it possible in the present scenario of genomics, and it's a very wonderful question, to induce safe mutations in the virus to reduce lethality? I mean, that's a remarkable question that has come up, but I would like to know.
Yes, so it is quite interesting question. So if you look at the data sets or the evidence which suggest attenuation of SARS-CoV-2, there are a set of genetic mutations which can attenuate SARS-CoV-2 and has been documented in literature. And these are genetic mutations and deletions, mostly stop gain mutations and deletions in a gene called ORF8. Now, incidentally, for the earlier SARS pandemic as well as for the MERS epidemic, there have been multiple reports of ORF8 deletions or ORF8 stop gain mutations which can attenuate virus. So it is a fact that there are mutations in SARS-CoV-2 which can attenuate the virus. Now, yes, theoretically in the lab, you can also make a SARS-CoV-2 with an ORF8 deletion. But the real point here is that then that is an attenuated virus, and then therefore will not propagate much easily or will not infect individuals as easily as probably the other isolates out there. So yes, in terms of creating a much better vaccine, probably yes. In terms of creating a strain to be distributed out there, probably no.
Doctor, we have one more question coming in from Dr. Rifa Khan, who's a young research scholar and has also done a lot of social medicine and work with the YRG group from Chennai. So she wants to know, how can public health practitioners and clinicians, and really, are there any tools which exist for them to sort of know how genomics is affecting the population at large in terms of COVID, and really what can be done from their end to understand the immune response?
Yeah, so the slide that I sort of displayed before, which is a phylogenetic tree. Now this tree is available on the URL down there. It's a browsable tree. So you can actually search through your state, in many cases your district, if there is enough number of sequences done for that particular state, and be able to understand what is the virus that is out there and predominantly out there. So it can provide a lot of information about the virus isolate, so on and so forth. And in tandem with that, what you also need to understand is, of course, for this you need to have some basic idea of the immune response, the immune epitopes, and so on and so forth. The earlier resource that I sort of mentioned, which is INDICOV, provides a lot of information about genetic variants in this isolates. So you could search particular isolate in this particular resource, and it will give you a set of information about where does the mutations occur in these isolates. So these are essentially two complementary resources. One is to look at the genetic lineages of the virus, and the second one is to dive deeper into each of these viruses to understand what does the genetic mutations contribute to in terms of functionality.
And is there any data set which is available or tools which are available to understand also the heterogeneity in immune response?
Uh, Frankly speaking, not yet. As I said, we really do not understand really much about how the immune system responds to the virus. And of course, what's the longevity of the immune response. There is enough evidence to suggest that both T cell and B cell responses are important, and that at least the B cell response doesn't seem to be quite long lasting, as suggested from very recent evidence and also from evidence on other coronaviruses out there. But we really do not understand the T cell responses yet to that level of granularity, because practically what we assay as a marker for immune response is antibody levels, which is essentially the B cell response.
Right. So to just sum up, sir, and we have probably a sum up question from a very senior professor, Dr. K.R. Antony, who is also the independent monitor of the National Health Mission in Kochi. So he would like you to sum up to tell us whether the genomic variants in India indicate any variation in terms of virulence, infectivity, and clinical course.
In terms of infectivity, yes, a significant proportion of the viruses belong to the D614G class, which is a A2A clade, which is much more infective. In terms of mortality, in terms of severity, until now, there is no evidence of a virus that can create more mortality or more severe illness, and then therefore there is nothing to suggest so. In terms of attenuation of the virus, which is essentially the OR of 8 deletions or stop gain mutations, uh, these mutations have occurred in India, and there are multiple viruses even within our sequences and sequences by others, which suggest that there are stop gain or deletion mutations which attenuate the virus. These still remain in very small numbers, and then therefore it's very unlikely to create a sort of a euphoria or a public health euphoria that you have an attenuated virus.